I was looking over the source material for today's deep dive into chapter 12, biotechnology and its applications, and I had this massive realization that completely shattered my preconceptions.
Okay, I love when that happens. Lay it on me.
Well, if you are approaching this topic for the first time, you probably have the exact same mental image I did. You hear the word biotechnology and you picture purely synthetic chemistry.
Oh, for sure. Test tubes, glowing liquids, that whole thing.
Exactly. You imagine scientists in sterile white coats, hunched over lab benches, using these little eyedroppers to meticulously mix chemical A with chemical B in a glass flask just to synthesize a drug from scratch.
Yeah, the classic Hollywood scientist trope.
Right. But the introductory text for this chapter makes it explicitly clear that this mental image is entirely backward. Biotechnology isn't about human beings inventing manufacturing processes from scratch. It is, and this just blew my mind, it is literally the hijacking of nature's own factories.
Hijacking nature's factories. That is a fantastic way to put it.
Yeah, because we are taking these optimized, microscopic manufacturing plants that nature has spent, billions of years perfecting, living cells, and we are just uploading our own genetic blueprints right into them.
That paradigm shift, that exact realization you just had, is the absolute perfect place to start. Moving from humans building molecules to humans programming life to build molecules for us. That is the core essence of this entire scientific field.
It's just wild to think about.
It really is. And for you listening, our mission today is to dissect the foundational framework of this subject. Because as someone who has mentored students through this specific Neet material for over 15 years, I can tell you something for a fact.
What's that?
Jumping straight into the complex mechanics of, say, recombinant DNA without understanding the broad conceptual roadmap, it is a total recipe for disaster.
Yeah, you just get lost in the weeds immediately.
Precisely. So, we're going to break down the exact definition, the three absolute critical pillars of research, the broad applications, and the strategic layout of the chapter. If you understand the blueprint we're laying out today, the immense complexity of the later material practically organizes itself in your mind.
That makes total sense. So I guess the first logical question to tackle is simply why? Like, why does this field even exist in the first place?
That is the perfect Socratic starting point. Why do we need it?
Right, because we have massive chemical manufacturing plants around the world. We have brilliant human chemists who can synthesize incredibly complex polymers and plastics and all sorts of things. So why do we need to bother with the messiness of biology at all? Why use living things to make our medicines instead of just building them in a traditional chemical factory?
It fundamentally comes down to a matter of thermodynamic elegance and just extreme structural complexity. Human chemistry labs, despite all our advanced technology and massive stainless steel reactors, they are astonishingly clumsy compared to a single bacterial cell.
Clumsy. Really? Even with all our modern tech?
Oh, incredibly clumsy. Think about it. If a human chemist wants to build a really complex three-dimensional therapeutic protein, they usually have to rely on brute force.
Meaning what, exactly?
Meaning extreme heat, immense atmospheric pressure, and highly volatile toxic solvents. And even with all of that aggressive chemistry, the yield of the final correctly folded protein might be incredibly low. Plus, you're producing a massive amount of hazardous chemical waste in the process.
Okay, wow. But a cell doesn't need any of that extreme stuff. It operates at just standard room temperature, right? And standard atmospheric pressure.
Exactly. A microscopic bacterial cell is an optimized manufacturing plant that performs thousands of complex, multi-step, localized chemical reactions simultaneously at ambient temperature.
That's insane when you really picture it.
It does this quietly, continuously, and efficiently without generating toxic byproducts that would, melt the factory down. It does all of this simply by reading the instructions coded in its DNA and translating them into proteins. Nature is, frankly, the greatest chemical engineer that has ever existed.
So biotechnology exists because human engineering basically hit a wall.
We hit a ceiling regarding molecular complexity, yes.
Yeah.
We realized we couldn't efficiently build these intricate molecules ourselves, so we decided to outsource the labor to the microbes.
We aren't competing with nature, we're co-opting it.
Co-opting it entirely.
Man, that makes the first sentence of the chapter incredibly significant. Let me pull it up here. The text defines biotechnology by stating that it essentially deals with industrial scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. Every single word in that definition feels loaded now.
It is very carefully constructed. Let's isolate the first defining phrase there, industrial scale. In the academic world, especially if you're tackling competitive exams like NEET, students often overlook this phrase entirely and focus only on the genetics part. Why do you think the text leads with the concept of scale?
Well, because finding a theoretical cure in a tiny petri dish doesn't actually solve a human problem. Like, if I manage to engineer a single bacterium to produce a new cancer-fighting protein, but I only have enough to fill a microscopic capillary tube, I haven't actually created a viable biotechnology.
You've created a neat lab trick, but not a solution.
Exactly. The word industrial dictates that this science is fundamentally tied to mass manufacturing. It's that massive leap from a laboratory discovery to producing millions of liters of a product for global distribution.
Well, let me tell you, the translation from the lab bench to the factory floor is the hardest part of the entire discipline.
Really, harder than the gene editing itself.
Often, yes. If a process works in a 10 milliliter test tube but completely collapses when you try to run it in a 100,000 liter steel vat, it is not biotechnology. It's just an experiment.
So scale is everything.
Scale is everything. And what exactly are we scaling up? The text specifies two distinct categories of products, biopharmaceuticals and biologicals.
Okay, yeah, I wanted to ask about that. Those terms sound nearly identical to me, but the text definitely treats them as separate entities.
They do, and you need to know the difference.
To me, a biopharmaceutical implies a direct medical application, like a pharmaceutical drug that is synthesized biologically rather than chemically. So things like synthetic insulin, vaccines, lab-grown antibodies. But biological sounds like a much broader umbrella term. Am I on the right track?
You are spot on. The distinction is subtle but critical for understanding the true scope of the field. Biopharmaceuticals are strictly therapeutic or preventative medicines intended for human or animal healthcare.
Okay, medicines. Got it.
Biologicals, on the other hand, encompass a vastly wider range of products. A biological could be an industrial enzyme used in your laundry detergent to break down stains.
Oh, wow. I didn't even think about detergent.
Right. Or it could be a specialized protein used to clarify apple juice in mood processing. It could be a biopesticide used in agriculture. By explicitly listing both terms, the text is warning the reader not to pigeonhole biotechnology merely as a branch of medicine.
So we have our massive industrial scale factories pumping out both life-saving medicines and everyday industrial products.
Exactly.
Okay, the second half of that core definition explains how we actually do this. It says, using genetically modified microbes, fungi, plants, and animals. This is where I started to build a mental map of the different tools at our disposal. Because the text doesn't just say bacteria, it lists 4 distinct types of living factories.
And in the academic world, this right here is a classic high-yield trap.
Oh, really? How
Yeah.
so?
Because the vast majority of introductory genetics focuses heavily on bacterial experiments using E. coli. Because of that, people develop severe tunnel vision. They equate the term genetically modified strictly with microbes. But you listening, you must retain the complete picture. Let's look at why all 4 of these categories are necessary, starting with those microbes.
Okay, well, I kind of think of microbes like bacteria as the fast, cheap 3D printers of the biotech world.
I like that analogy, 3D printers.
Yeah, because you give them a relatively simple genetic blueprint, and since they multiply incredibly fast, I mean, some double their population every 20 minutes, right?
They do. It's exponential growth.
Right, so they can churn out massive quantities of basic proteins super quickly. They are simple, they're efficient, and they're really cheap to maintain.
They are the undisputed foundational workhorses of the industry. Bacteria are prokaryotes, meaning they lack a complex nucleus and they don't have membrane-bound organelles. Their internal machinery is very straightforward. If you need a simple, unmodified protein sequence, they are absolutely perfect.
But there's a catch, I assume.
There is a major catch. Their simplicity is also their limiting factor. Many of the most crucial human proteins are highly complex. After a human cell builds a protein, it often modifies it. It might add complex sugar chains in a process called glycosylation, or it might fold it into highly intricate, specific 3-dimensional structures.
And a simple bacterial 3D printer just doesn't have the hardware to do those complex finishing touches.
It simply doesn't. If you force a bacterium to make a complex human protein, it often misfolds it, creating this useless, tangled clump of amino acids inside the cell.
Ah, so it makes the raw material but can't assemble the final product correctly?
Exactly. That is why we must step up to the second factory on the list, Fungi.
Okay, so if microbes are the fast 3D printers, fungi like yeast are the slightly more sophisticated assembly lines.
That's a good way to bridge it.
Because fungi are eukaryotic, right? They actually have that complex internal cellular machinery so they can handle manufacturing proteins that require a little more post-production modification.
Spot on. Fungi bridge the gap. They grow relatively quickly and cheaply in large vats, much like bacteria do, but they actually possess the advanced endoplasmic reticulum and Golgi apparatus required to perform those complex protein modifications.
Okay, that makes sense. But what about plants? The text lists plants third.
Sometimes even yeast isn't enough, or the sheer volume required is just too massive for a traditional steel factory. That brings us to plants.
Plants completely change the manufacturing paradigm for me. I view plants as the ultimate solar-powered outdoor factories.
Solar-powered outdoor factories, that is brilliant.
Well, think about it. If you need to produce a biological product on a truly massive global agricultural scale, building a $1 billion stainless steel bioreactor facility just isn't economically viable.
Not at all. The overhead would be astronomical.
Right. So instead, you can genetically modify a crop. The sun provides the infinite energy, the soil provides the raw elemental materials, and the plant's own biology does the manufacturing right there in the open field.
The economics of plant-based biotechnology, which is often called molecular farming with a P, are just staggering. You're leveraging the Earth's natural energy cycles to manufacture complex biologicals.
So we're talking about, like, what kind of crops?
You can engineer tobacco plants to produce human antibodies, or you can modify corn to produce industrial enzymes right inside its seeds. The bioreactor is literally just an open field.
That is wild. And then finally we arrive at the most complex category on the list, animals.
Yes, animals represent the highly specialized, ultra-complex manufacturing hubs.
So why would we need an animal? Is it for the most difficult stuff?
Exactly. If you need a biological product that requires the absolute most intricate folding and post-translational modifications, something that only functions if it's produced in a fully mammalian system, you have to use an animal.
Do you have an example of that?
A classic example is engineering a transgenic goat so that it secretes a vital human therapeutic protein directly into its milk.
Wait, medicine in goat milk?
Yes. The mammary gland of a transgenic mammal is arguably the most sophisticated bioreactor on the planet. Extracting a complex pharmaceutical from milk is incredibly efficient.
That's a mind-blowing realization right there. Medicine from milk.
It is incredible. So the syllabus lays out this specific progression for a reason. Here's what you must remember. The textbook explicitly includes microbes, fungi, plants, and animals as genetic hosts. It is a hierarchy of manufacturing complexity, moving from the simple, rapid replication of bacteria all the way to the highly complex protein secretion of mammalian systems.
Okay, got it. Having these 4 distinct types of living factories is incredible, but a factory is totally useless if you don't know how to run it.
Very true.
Which brings us to the absolute core of the chapter's introduction, the 3 critical research areas of biotechnology.
Yes. To master this subject, memorizing isolated facts is insufficient. You need a structural framework. The text outlines 3 critical research areas which we can translate into the CCD framework.
CCD, catalyst, conditions, downstream.
Exactly. Every single breakthrough, every patent, and every engineering challenge in biotechnology research falls into one of these 3 pillars.
Okay, let's unpack pillar 1, the catalyst. The text states that the first critical research area is, quote, providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme.
Notice the specific vocabulary there.
Yeah, the use of the word catalyst is fascinating here, because in a standard high school chemistry class, a catalyst is usually like an inorganic metal, like platinum, used to speed up a reaction.
Right, the text uses that specific term to deliberately bridge chemistry and biology. In a traditional chemical factory, a catalyst lowers the activation energy of a reaction without being consumed itself. But in our biological factory, the genetically modified organism is the machine driving the chemical conversion.
So we feed the organism and it does the work.
Yes, we feed it raw nutrients, sugars, nitrogen, basic salts, and its internal biological pathways act as the catalyst to turn out the biopharmaceutical.
So the entire first area of research is dedicated to finding or aggressively engineering the absolute best worker for the job.
The perfect employee.
Yeah, you might start with a wild strain of yeast, but you spend years mutating it, editing its genes, and deleting wasteful metabolic pathways until it becomes this hyper-efficient catalyst dedicated solely to producing your specific target molecule.
You are systematically removing biological inefficiencies to create that perfect worker. However, look closely at the exact phrasing of the text again. It states the catalyst is usually a microbe or pure enzyme.
Or pure enzyme.
This is a crucial distinction. Sometimes using a whole living cell is incredibly messy.
Messy how?
Well, living cells need constant feeding, they produce their own waste, they can get sick, and they can mutate or die off.
Ah, right. They have their own agenda.
Exactly. So instead of dealing with a whole living factory, you just extract the specific machine you need from inside it. Scientists will often isolate and purify the specific protein machinery, the enzyme, that performs the desired chemical reaction.
So they just rip the engine out of the car.
That is a perfect analogy. They discard the rest of the cell and use only that pure enzyme as the catalyst in the reactor. It's cleaner, it's easier to control, and it vastly simplifies the purification process later. So here's what you must remember. The catalyst in biotech is not always a whole living organism. It can be an isolated, pure enzyme.
Okay, that makes Pillar 1 incredibly clear. You secure the catalyst, either the whole improved organism or the pure enzyme. But having the best worker in the world doesn't mean anything if the workplace environment is completely chaotic.
A terrible factory floor ruins a great worker.
Exactly, and that transitions us into Pillar 2, optimal conditions. The text defines this as, quote, creating optimal conditions through engineering for a catalyst to act.
The key word in that definition is engineering. What does that word suggest to you about our relationship with these organisms?
It suggests complete active control. We aren't just putting a genetically modified microbe in a glass jar on a windowsill and hoping nature takes its course. We are aggressively designing a highly artificial environment.
We are playing God with their environment, yes.
Because if we have this super worker catalyst, but the factory floor is too hot, too acidic, or suffocating due to a lack of oxygen, the worker stops producing or dies.
This is where biology forcefully intersects with physics, thermodynamics, and fluid dynamics. In nature, a microbe experiences wild fluctuations in its environment. Temperature drops, nutrient scarcity, competition from other microbes. It has evolved to survive, not to overproduce.
Right, survival of the fittest, not survival of the most productive.
Exactly. But in an industrial bioreactor, survival isn't enough. We demand maximum yield, so we create highly controlled artificial utopias for them.
I picture these massive stainless steel bioreactors, sometimes holding tens of thousands of liters of liquid. The engineering required to maintain a utopia at that scale must be just mind-boggling.
The physical challenges of scale-up are immense. Consider just the temperature, microbes metabolizing sugar-release heat.
Like when you work out and get hot.
Exactly like that. In a small flask, that metabolic heat easily dissipates into the air in the lab. But in a 50,000-liter tank, the volume to surface area ratio is vastly different. The heat is trapped inside the core of the liquid.
Oh, wow. I didn't think about that.
If we don't actively engineer immense cooling jackets to circulate chilled water around the outside of that tank, the microbes will literally cook themselves to death in their own metabolic heat.
That is terrifying. So you have to constantly monitor and adjust the cooling.
Constantly. And it's not just temperature either. It's pH levels. It's ensuring the liquid is perfectly mixed so the nutrients don't just settle at the bottom. It's pumping in sterilized oxygen for aerobic bacteria.
How do you even mix 50,000 liters of thick biological soup without damaging everything?
It's a delicate balance. We use massive mechanical impellers to stir the broth, but if they spin too fast, the sheer stress will physically rip the cellular membranes apart.
Oh, you basically blend them.
Yes, you'd puree your own workers. And we use spargers to bubble oxygen through the liquid, but if it creates too much foam, it blocks the exhaust filters and pressure builds up. Research Area 2 is entirely about mastering these massive physical variables to maintain that perfect artificial utopia where the organism has absolutely nothing else to do but produce our target product.
It's like building the ultimate luxury suite for a hyper-focused workaholic. Perfect room temperature, endless room service, optimally filtered air. Just keep working.
That is exactly what it is.
But that leads to the final and perhaps most overlooked step. We have Pillar 3, downstream processing. The text defines it as, quote, downstream processing technologies to purify the protein/organic.
Listen to me very carefully on this one.
Yeah.
This is the most critical juncture in the entire process, and it is the single biggest trap for students learning this material.
The biggest trap. Really?
Yes. People become incredibly enamored with the upstream processes. They love learning about recombinant DNA, CRISPR, gene splicing, and the massive high-tech bioreactors. They focus entirely on the creation of the product.
It's the sexy part of the science.
Exactly. And because of that, they completely forget that at the end of the fermentation cycle, you don't just magically have a neat, clean vial of medicine sitting there.
Right. You have a massive industrial-sized vat filled with an incredibly complex biological soup. I like to think of it like a river.
A river. Walk me through that.
Well, upstream is where all the construction and manufacturing happen. But downstream, where the river ends, you have this chaotic mix. You have millions of dead and dying cells, leftover sticky nutrient broth, toxic metabolic waste products, and floating somewhere inside that massive sludge is your tiny, highly fragile target protein.
That is a brilliant visualization. And let me tell you, if you were to take that raw upstream sludge and inject it into a human patient, the results would be catastrophic.
It would probably kill them.
It would. The human immune system would violently attack the bacterial debris, leading to severe anaphylactic shock. The biological product is entirely useless until it is isolated from the sludge.
And that is the essence of downstream processing. It is the arduous art of purification and separation. It's filtering the river water.
Yes.
You have to figure out how to separate the intact cells from the liquid broth, crack the cells open if your protein is trapped inside them, extract the specific molecule you want, and purify it to an exact clinical standard. And you have to do all of that without accidentally destroying the fragile protein in the process.
Downstream processing often utilizes highly advanced separation techniques. We use massive industrial centrifuges spinning at thousands of RPMs to force the heavy cells to the bottom of the tank. We use specialized filtration membranes that only let specific molecule sizes pass through. We use advanced chromatography columns that act like chemical sieves, capturing only our target protein while letting the rest of the waste wash away.
It sounds like finding a microscopic needle in a 10,000-liter haystack and doing it without breaking the needle.
It is arguably the most expensive and technologically difficult part of the entire biotechnological process.
Seriously, the purification is the hardest part.
Without a doubt.
Yeah.
You can engineer the most brilliant microbial catalyst in the world, and you can build a flawless $100 million bioreactor to provide the optimal conditions. But if your downstream processing technologies fail to purify the final protein efficiently, the entire venture collapses.
Wow.
So here's what you must remember. Downstream processing is unequivocally synonymous with purification and separation. It is the inescapable final step before any product goes to market.
Okay, the CCD framework makes the whole process so logical now. Catalyst is the genetically engineered worker. Conditions is the massive, highly engineered factory floor. Downstream is the rigorous quality control, purification, and packaging at the end of the line.
You've got it perfectly.
So now that we understand the mechanics of how biotechnology operates, we need to examine what it is actually accomplishing in the real world. The text casts a remarkably wide net of applications.
It does. The explicit list provided in the text is essentially a rapid-fire overview of human problem-solving. It mentions therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. We need to categorize these to understand their impact.
Right. Well, the text highlights that biotechnology is used, quote, especially in the field of food production and health. Let's start with health, which is divided into therapeutics and diagnostics.
Okay. How do you see the difference between the two?
Well, the distinction between those two is fundamentally a sequence of medical care, right?
Exactly. Diagnostics is the act of discovery. We are using biotechnology to find the disease before it destroys the body. Instead of waiting for a patient to show severe physical symptoms, we use highly sensitive diagnostic tools.
Like PCR tests, right?
Yes, polymerase chain reaction, or PCR, which amplifies tiny trace amounts of DNA so we can detect a virus early, or ELISA tests, which use specialized engineered antibodies to detect specific viral proteins in a blood sample. Diagnostics allow us to see the invisible.
And once we've found the problem, we pivot to therapeutics, which is the act of fixing the disease.
Precisely. Therapeutics are the biopharmaceuticals we discussed earlier. It is the application of our CCD framework to produce a medicine.
Yeah.
A perfect historical example of this is human insulin.
Oh, the insulin story is wild.
It really is. For decades, diabetics relied on insulin extracted and purified from the pancreases of slaughtered pigs and cows.
Which just sounds so inefficient.
It was. It took massive amounts of animal tissue to get tiny amounts of insulin. And because pig insulin wasn't perfectly identical to human insulin, some patients developed severe allergic reactions over time.
But biotechnology completely revolutionized that. Scientists isolated the actual human gene that codes for insulin, inserted it into a simple E. coli microbe, our pillar 1 catalyst, and grew them in massive bioreactors.
Yeah.
Suddenly, we weren't harvesting animal organs. We had bacterial factories churning out vast quantities of pure, authentic human insulin.
That is the ultimate triumph of therapeutics. It solves a massive supply issue while simultaneously increasing the safety and efficacy of the drug. Now, let's move from the hospital to the agricultural fields. The text lists genetically modified crops and processed food.
This represents a huge shift in the application of the technology. We are moving from engineering microbes in steel tanks to engineering the plants themselves.
We use the exact same foundational genetics, but we apply it to our food supply.
We might modify a plant to be inherently resistant to a specific insect pest, drastically reducing the need for chemical pesticides, right?
Yes, like bright cotton.
Or we might modify the nutritional content of a crop to fight global malnutrition, like golden rice.
The impact here is measured in billions of lives. By modifying the genetic makeup of agriculture, we are attempting to secure the global food supply against climate change, pests, and resource scarcity. But the list in the text doesn't stop at health and food. It transitions into applications that are often entirely overlooked by the general public.
And this brings me to what I consider the most fascinating pivot in the text. When I read the words bioremediation and waste treatment, it genuinely expanded my view of the entire field.
It's a huge shift in perspective.
Because we spend so much time talking about biotechnology as a manufacturing tool, right? Making things for humans to consume. But bioremediation treats biotechnology as an erasure tool. It's deploying living cleanup crews to fix our industrial mistakes.
The word itself is highly descriptive. Bio meaning life, remediation meaning to remedy, fix, or cure.
Right. So consider an environmental disaster, like a massive crude oil spill in the ocean. Traditional human methods for cleaning it are slow, mechanically clumsy, and often involve dumping harsh chemical dispersants into the water, causing even further ecological damage.
It's often just sweeping the dirt under the rug.
Exactly. But bioremediation asks us a different question. What if we engineer a microbe whose optimal environment is an oil slick?
It's brilliant. We take the CCD framework and deploy it in the wild. We find or modify a bacterial catalyst whose natural metabolic process literally digests toxic hydrocarbons.
It eats the crude oil.
It eats it, breaks it down for energy, and excretes harmless byproducts like water and carbon dioxide.
It is the ultimate application of biological scale to combat industrial scale destruction. That just blew my mind.
It's ecosystem restoration on a microscopic level. And waste treatment operates on a similar, albeit more controlled, principle. Every major city on Earth relies on massive biological reactors, often called activated sludge processes,
Where complex communities of microbes break down human sewage and industrial organic waste, purifying the water before it is released back into the rivers and oceans.
We are quite literally unleashing microscopic heroes to clean up the planet. And right next to that environmental focus on the list is energy production.
Which completely bridges the gap back to the industrial scale definition for me. We aren't just making tiny vials of targeted medicine, we are using microbes to generate literal fuel to power our infrastructure.
Biological energy production is a massive area of research. We can utilize specific microbes, like methanogens, to digest organic waste, from agricultural runoff to municipal garbage, and capture the methane gas they excrete as a clean-burning fuel.
Or we can engineer advanced strains of yeast to rapidly ferment complex plant sugars into bioethanol to run our vehicles. We are hijacking the biological processes of digestion and fermentation to power our cities.
The applications are incredibly vast, from curing diseases to altering crops to cleaning oil spills to powering cars. But because the list is so diverse, I can see how it would be tricky to keep straight during an exam.
Yeah, there's a lot to memorize there.
And examiners know that. In an academic testing environment, they love this specific sentence. They will create a multiple-choice question listing 3 genuine applications, for instance, therapeutics, bioremediation and diagnostics, and then they will slide in a fake, vaguely scientific-sounding term, like telemetry or metallurgy. And they will ask, which is not an application of biotechnology according to the text. If you haven't internalized the exact boundaries of this list, it is very easy to second-guess yourself under pressure.
Right, because metallurgy sounds like it could be related to enzymes breaking down or something if you aren't sure.
Exactly. So here's what you must remember. The core applications explicitly defined are therapeutics, diagnostics, GM crops, processed food, bioremediation, waste treatment, and energy production. Lock those 7 terms in.
Okay, that gives us the entire conceptual foundation. We know it's about scaling up biology. We know the 4 factories: microbes, fungi, plants, and animals. We understand the 3 pillars:
Yeah.
catalyst, conditions, and downstream purification. And we have the broad map of applications.
That is the foundation, yes.
So where does this introductory text ultimately point us? What is the roadmap for the rest of Chapter 12?
The text provides a clear navigational outline at the end of the introduction, mapping out 4 specific subsections that the student will journey through. Section 12.1 focuses on biotechnological applications in agriculture. Section 12.2 covers biotechnological applications in medicine.
The big 2.
Right. Then Section 12.3 details transgenic animals. And finally, Section 12.4 tackles ethical issues.
The structure makes perfect sense based on everything we've discussed. Sections 1 and 2 cover food and health. Section 3 dives deep into the most complex and advanced of our 4 living factories, transgenic animals. But it's that final section, 12.4 ethical issues, that really forces you to pause.
It does. It stands out from the hardcore science.
Yeah, it feels like an inevitable high-speed collision between raw scientific capability and human morality.
It is a profound inclusion in a science text. Why do you think a hard science textbook dedicated to the rigorous mechanics of molecular biology dedicates an entire final section to ethics?
Because of the sheer magnitude of the power being discussed. I mean, think about the capabilities we just mapped out. If we master the 3 pillars, if we can design any genetic catalyst, engineer perfect industrial conditions, and purify the result across all 4 domains of life, we essentially possess a control panel for the biological operating system of the planet.
A control panel for life. That's a heavy thought.
We have the unprecedented power to rewrite the genetic code of living organisms to serve human industrial needs. When you hold that kind of power, a section on ethical issues isn't just filler text at the back of the chapter. It is the crucial philosophical brake pedal on the scientific gas pedal.
It is the recognition that understanding how to manipulate life does not inherently answer the question of whether we should. The syllabus requires students to study these ethical boundaries just as rigorously as they study the biochemical pathways. We're talking about transgenic manipulation, taking a gene from a human and putting it into a pig, or taking a gene from a bacterium and putting it into a food crop.
And once you release a genetically modified organism into the open environment, like a field of transgenic crops, you can't exactly put it back in the test tube if something goes wrong. It becomes part of the global ecosystem forever.
Exactly. The chapter delves into the necessity of regulatory bodies to manage this risk. For instance, in India, the syllabus specifically highlights the GEAC.
What does that stand for again?
The Genetic Engineering Appraisal Committee. They make decisions regarding the validity of GM research and the safety of introducing GM organisms for public services. The text also touches on complex global issues like biopiracy, where multinational companies patent genetic resources or traditional knowledge from developing nations.
That sounds highly controversial.
It is. Now, we don't take a political stance here, and neither does the exam, but the text demands that you understand the profound weight of this technology. The power to cure genetic diseases and feed billions of people is inextricably linked to the power to permanently alter the genetic heritage of the Earth.
It perfectly contextualizes the entire discipline. It starts with a dry, technical definition of industrial-scale production, and it ends with the heaviest, most consequential philosophical questions facing modern science.
Which is exactly why building this conceptual map first is so vital. Before we conclude, it is time to lock this framework into your long-term memory. We're going to do a rapid-fire sticky summary.
All right, let's do it.
If you take nothing else away from this deep dive, these are the facts you must retain to navigate the rest of the material successfully.
Give me the absolute core takeaways.
Number 1, the defining shift is the word industrial. Biotechnology is fundamentally about massive industrial-scale mass production, not just small-scale laboratory discovery.
Got it. Scale is everything.
Number 2, the text explicitly lists 4 distinct genetic hosts. Here's what you must remember. It is microbes, fungi, plants, and animals. Do not fall into the trap of only remembering bacteria.
Microbes, fungi, plants, animals, the 4 factories.
Number 3, The entire field is built on the three CCD pillars. Pillar 1 is the catalyst, the engineered worker, which can be an improved organism or a pure isolated enzyme. Pillar 2 is conditions, the highly engineered artificial utopia where we control the thermodynamics and fluid dynamics to maximize production. Pillar 3 is downstream processing.
And downstream is the trap.
Which brings me to number 4. Here's what you must remember. Downstream processing is always synonymous with purification and separation. It is the difficult, inescapable final step of extracting the target product from the biological sludge.
Filter the river water.
Exactly. And finally, number 5. The applications are vastly broader than just medicine. They span therapeutics and diagnostics, agricultural crops, environmental bioremediation, waste treatment, and massive energy production.
That structural map makes the entire topic feel so approachable now. It's no longer just a random assortment of genetics and chemistry. It is a highly organized, sequential industrial process.
You now have the absolute perfect mental scaffolding to tackle the intricate details, the complex vocabulary, and the specific case studies that fill the rest of the chapter.
This has been incredible. As we wrap up this deep dive, I want to leave you, the listener, with a final provocative thought to mull over as you continue your studies.
I love a good thought experiment.
We talked extensively about those first two pillars, stripping away an organism's natural environment, rewriting its DNA from scratch to serve our specific industrial needs, and locking it inside a hyper-optimized steel bioreactor where its entire existence is dedicated to manufacturing a single human chemical.
Right.
At what point on that journey do we stop calling it a living creature and start calling it biological machinery? Keep that blurry line in mind as you dive into the rest of Chapter 12. Thanks for exploring with us.